So we will continue from where we left. I was just mentioning that this is the primary active transport, that the proton, they are directly transport from inside to outside by using the ATP, which is called as the primary active transport, and the this gradient developed by this primary active transport is utilized for the further movement of ions and sucrose uh, across the membrane. So this is called as secondary active transport. Solute transport across the biological membrane is very important phenomena, especially when we talk about the nutrient uptake. So there are three things which we will discuss here very briefly. The dynamics of solute transport, energy demand for ion uptake, and the kinetics of solute transport. So that will be discussed in the next upcoming slides. So transport proteins of transport protein of tonoplast and plasma membrane are mentioned in here in this cell diagram. So you can see here there are a lot of pumps and a lot of channels and transporters in a cell. So they are present at the level of the plasma membrane, and also they are present at the level of tonoplast. You can imagine that how complicated it is. That a multitude of secondary active transporters are present in the plasma membranes of root cells, which couple H influx to the movement of solutes against their electrochemical gradient. So you can imagine that the H ion gradient, which is developed by proton pumps, and that is utilized for secondary active transport, and that is used by plenty of transporters. You can imagine here at the plasma membrane level, nitrate H, Ht2PO4H, sulfate H, chloride H, then potassium H. And the similar you can see here in the tonoplast, even the boron H. So it means the H ions gradient which is utilized, which is developed by the proton pump, that is utilized by a plenty of uh, transporters. So it means this is not very simple that one proton is released from the proton pump and then that is used by the potassium. So this is a complex system. Proton-coupled transporters in the plasma membrane of root cells are responsible for the uptake of anions such as nitrate, phosphate, sulfate, and chloride. In addition, proton potassium importers such as those encoded by KUPHAK gene. So, so this movement of ions is not only of the cation but the anions as well. As mentioned, nitrate, phosphate, and then the sulfate, and even the chloride. So they are also dependent on this gradient which is developed by these proton pumps. And also the cations, potassium and the other cations, they are also dependent on the HI. So the movement or the influx or efflux through the plasma membrane or the biological membranes is different. It's not similar for all the ants. And the one thing which must be remembered, that this movement is not only one-sided, that we say that only the potassium, they are moving from outside to inside. At the same time, they are moving in and they are moving out, because this is a system which is collaborated with the outside, the system outside, that is basically the soil solution in case of the field plants or the nutrient medium. So this is a continuous process कि पोटेशियम अंदर आ रहा है, पोटेशियम बाहर जा रहा है, लेकिन जो इनफ्लक्स हमने मेजर करना है वो है नेट इनफ्लक्स, कि अगर 10 अंदर जा रहे हैं, 5 बाहर जा रहे हैं, तो 5 हमारे पास नेट इनटेक। तो आप ये देखें कि 5 इन आयंस के लिए 10 का अंदर आना जरूरी है और 5 का बाहर जाना जरूरी है। So again, it makes the system more complex. Very often, though, only the net uptake of ion is determined experiment. So which we determine कि हमारे कितनी पोटेशियम या कैल्शियम अपटेक हुई है, हम तो वही करते हैं जो नेट है, और जितनी वो मूव हो के अंदर आई फिर रिफ्लक्स हो के बाहर चलेगी, वो तो किसी खाते में नहीं है। So which is the net result of influx and efflux across the plasma. तो जो जो डायनामिक्स हैं उसके, so they are also dependent on that. Efflux can become similar in magnitude to influx, particularly at extreme low or extreme high concentration. जब बाहर एक्सट्रीम हाई या लो हो जाएगी कंसंट्रेशन, तो फिर ऑलमोस्ट वो इफ्लक्स और इनफ्लक्स सेम हो जाता है, क्योंकि उसकी जरूरत नहीं क्योंकि जो लो वेरी कंसंट्रेशन हो या हाई कंसंट्रेशन हो, तो उसके डायनामिक्स पे चेंज हो जाता है. It is also noteworthy that at a given external concentration, the efflux of a particular mineral nutrient can be many times higher from the roots of plants sufficiently supplied than from roots of the deficient plant. So this is basically the conclusion of this slide, that the, the movement of ions is dependent on the external concentration. So if you have a high concentration, so then the movement is different. When you have a low concentration, then the movement is different. So it means uh, the movement of ions is dependent on various factors. For example, as I mentioned earlier, the type of the charge, and then the external concentration, and the type of ions, whether as a cations or anions. And of course, there are also other factors like the proton concentration outside, the efficiency of proton pumps. If the proton pump are not efficient, then the movement of these ions will also be affected. The efflux of ions and other solute is affected by several factors: integrity of plasma membrane, the presence of transporters uh, in the protein, because the the number of transport proteins per unit area of plasma membrane also determine the movement of ions. Then the electrochemical gradient driving force for transport, uh, which I mentioned earlier, that that is basically the activity of proton pumps and the concentration of the solute in the cytoplasm. Inside वो sufficient है या deficient. अगर जो साइटोप्लाज्म के अंदर किसी आयन डेफिशिएंसी है, तो देन द मूवमेंट आयन विल बी डिफरेंट, और अगर वहां पे सफिशिएंट है, तो देन द मूवमेंट ऑफ आयन विल बी डिफरेंट. So these are basically four uh, factors which I mentioned in this slide, which are responsible for the dynamics of ions through the membrane. So this figure we have already mentioned, explained, and has also been explained. So basically, these tells us about the dynamics, that the movement of ion is not a very simple, it is a very complex phenomena, but it is moving very smoothly. Then energy demand, because we have mentioned many times that it depends on the movement of H ion from inside to outside, will develop the chemical gradient. So it means that also require energy. Here there is an example: respiratory energy cost for ion uptake in roots of Corex dendra. This is a plant species. Ion uptake and the plant age, growth, maintenance of biomass, proportion of total ATP demand required for. So when the plant age is 40 days, then ion uptake is more important. It means the more proportion of ATP is used for the ion uptake. And the growth is also important at that stage, then that is 39. And the maintenance of biomass, that is 25. Because here there is a more growth, more on growth. The emphasis is more on growth. It's not on the maintenance biomass. Then at 60 days, then the ion uptake is reduced, mean that it is not, its plant is a bit mature, the growth is relatively lower, and the ATPs are basically used to maintain and to promote the growth. And the basically the reservoirs uh, use in that the nutrient reservoirs which are present in the soil, sorry, in the plant cells, so they are used for the growth. Because in the start there is a very fast uptake, and then these are stored in the vacuoles as well. And then when the plants they are grown, the ions from the leaves they are moving toward the upper part, the growing part, and to the reproductive part. So it means the inside movement is then more important as compared to the ion uptake through the roots. Then on later stage again it is reduced because the ion uptake is lesser, ion requirement is lesser at the later stages, and these are the component, the other components. So it means for ion uptake in the start, early plant growth, young age, so they require more energy as compared to the older age. In cereals, 76% of respiratory energy has been reported for low affinity nitrate flow through plasma. So then the nitrate, as I mentioned earlier, that this is transported through along with the protons. So if you have only nitrate source, so they will use very much respiratory energy for the transport. Then, as I mentioned earlier, that the movement is not only inside the cells, there is also efflux and influx. So this is a ratio, and this is a concentration in the rhizosphere. So when we have a low concentration, as I mentioned earlier, अगर पिछली स्लाइड में देखें, मैंने कहा जब वेरी लो कंसंट्रेशन होती है तो इफ्लक्स और इनफ्लक्स जो है वो उनमें जो वेरिएशन जो है वो ऑलमोस्ट जीरो हो जाती है। लेकिन जब वो कंसंट्रेशन बढ़ेगी राइजोस्फीयर में तो वो उसका इफ्लक्स टू इनफ्लक्स रेशियो ज्यादा हो जाएगी, क्योंकि इफ्लक्स फिर ज्यादा, इनफ्लक्स भी ज्यादा होगा लेकिन इफ्लक्स फिर ज्यादा होगा। एंड देन जब वो हाई कंसंट्रेशन पे, वेरी हाई पे चली जाएगी, उसके बाद फिर वो स्मूथ, ये यहाँ से आगे जो है ये काइंड ऑफ स्मूथ लाइन है पैरेलल, तो फिर वो जो वेरिएशन है वो वेरी हाई कंसंट्रेशन वो लो हो जाएगी। तो इट मीन्स के जो ये फ्लक्स टू इनफ्लक्स रेशियो बढ़ रही है, तो यहाँ पे इसका मतलब है कि ये जो एनर्जी यूज हो रही है ये फ्यूटाइल, तो जितना इफ्लक्स ज्यादा होगा, क्योंकि इनफ्लक्स पे तो एनर्जी लग गई, इफ्लक्स पे भी एनर्जी लगती है टू सर्टेन एक्सटेंट, लेकिन वो दोबारा फिर इनटेक उसकी होगी, इसका मतलब वो एनर्जी जो लगी है उसकी इनटेक पे जो बाहर निकल गया, इफ्लक्स हो गया है, तो वो तो फजूल में गई। तो दैट इज बेसिकली द एनर्जी विच इज यूज्ड फॉर द फ्यूटाइल मूवमेंट, और वो उस वक्त ज्यादा होगी जब हाई कंसंट्रेशन उसके प्लांट्स के अंदर, सॉरी प्लांट मीडियम के अंदर हाई कंसंट्रेशन होगी राइजोस्फीयर में। सो So it means the energy demand is not only for the ion intake, but also the futile movement of ions required. The last that is the kinetics absolute transport. That is very simple, which is mostly used for the enzyme. That is Michaelis-Menten equation. And this is used to calculate the energy of the, to understand the kinetics of the slow transport. So this is the formula you might have known very many times, uh, because most of you are biological students, science students. So they mostly have this formula for their enzyme activities. This is Vmax as mentioned here, then the Km value when at level of half Vmax. And by using this formula, um, here you can also see that the external concentration has also been mentioned here, because that will also affect the transport rate. So this formula is used for the energy transport. So there are few example which has been mentioned here. You can see here the kinetics absolute transport, boron supply, boron concentration. Then uh, there are two different varieties. You can just see to have a look at. So this was all about the uptake of the nutrients of ions or ions from the nutrient medium, and you can ask if you have any question. Thank you very much.